Morning, Ellis students. I am not going to be in your biology class today, so I pre-recorded a lesson for you. Please go through and watch this video. As you are watching this video, I will cue you to pause periodically for you to work on some practice problems in your packet. Um, and this will kind of help you work through some of these problems. And then after you're done pausing and working on it, I will show you how to solve it. But that's just kind of our little what we're doing today. So today we're going to be thinking about non-Mendelian inheritance. If you remember before break, we did a lot of crosses like this one right here, where we crossed two homozygous individuals together, in this case peas. These are like Mendel's peas. Mendel crossed white peas and purple peas, and what he found that was really interesting was that all of the peas were purple and kind of looked like one of the parents. And Mendel was like, ooh, something must be dominant. And that works most of the time. But what's interesting is that if Mendel would have picked a different plant, he probably actually never would have made the discoveries he did. But what's interesting is that since Mendel's time, we've done crosses and learned different things. And this one's kind of interesting. You cross two homozygous snapdragons, a red snapdragon and a white snapdragon. What you might expect to see is either red or white. What you actually see is pink. Okay, so a little bit different. You cross two homozygous 4 o'clock flowers, a pink 4 o'clock with a white 4 o'clock. What do you think you're going to get? Okay, based on the previous one, you might say pink or white or light pink, but actually the offspring are pink and white. So the big idea here is that all of these plants have one gene that determines color. Okay, so we're not looking at a multiple gene situation here. They're all one gene. The genes are going to code for the expression of proteins, in this case pigments, and all the plants have two alleles for this gene. So the way that these alleles are expressed is just a little bit different, and that's what we're going to spend some time today looking at. So we've already looked at simple dominance. Only one dominant allele is needed to express. So you cross white and purple, all the offspring are purple, and we're really comfortable with how to do that. Today we're going to start with incomplete dominance. With incomplete dominance, just as I showed you before with our snapdragons, you cross a red snapdragon and a white snapdragon, and when you look at the offspring, they happen to be all pink. What do you notice about how the offspring compare to the parents? If you think about red and white, okay, where is pink at? Pink is kind of that intermediate phenotype of those two parents. So we're seeing a really interesting pattern here. And we call this incomplete dominance. And what's really interesting about incomplete dominance is that the phenotype of the individual with the heterozygous genotype are the intermediate of the homozygous genotypes. So if I look here, okay, what does that mean? Okay, my individual that is red, okay, and my individual that is white, okay, my heterozygous individual that has one red allele and one white allele is going to be an intermediate. It's halfway, it's kind of like in between. And kind of a good way to think about that is this individual is expressing one of these red alleles and is not expressing this white allele. So white is like almost like the absence of pigment. And so when you look at this this pink snapdragon, it really just has half as much red pigmentation, and half as much red pigmentation looks pink. So if you looked at the pigment protein that's being produced, it would be identical to that of the red. There's just half as much of it. So one allele is expressed and the other allele is not expressed in this condition. Two alleles are expressed more than one allele in the heterozygous genotype. So, and so when we look at our red, it's expressing 2 times as much red pigment because there's two alleles that are each coding for red pigmentation. The pink one has one allele coding for red pigmentation, and because there's half as much red pigment, it looks pink. And then the white one has no red pigmentation. And what's cool is that if you cross one of these, if you take these pink flowers and you cross them, you actually get in the F2 generation, you'll get some that are red, okay, some that are pink, and some that are white. and we see this really unique 1:2 to 1 ratio that is pretty signature of incomplete dominance. So we're going to look at Wendy the Whippet. If you notice anything about Wendy, she is super jacked. And we're going to be looking at a cool gene called the myostatin gene. I have a funny video that I will show you in class when we meet again. It's just not going to play on my player. So I will show you Wendy the Whippet when we meet together. But I would like for you to take a couple minutes and see if you can do some practice problems in your packet. The gene that we're going to be focusing on is myostatin, and it regulates muscle growth. So this is a regulatory gene. And what we've noticed is that non-racing whippets, okay, so those are like really, really like little tiny ones, they have two normal myostatin genes, so big M, big M. And they happen to be like extra little tiny, like 30-pound dogs. Racing whippets, and these are actually used in dog racing, are heterozygous for the myostatin gene. So they have one functional and one unfunctional copy of the myostatin gene. So the one functional copy regulates muscle growth. The unfunctional copy does not. And because the muscle growth is not regulated, they get a little bit more muscular. And there's a picture of one of these right here. And those muscular whippets can just run a little faster, but still really aerodynamic. This is something we call a heterozygous advantage. And then the bully whippets, which is what Wendy is an example of, have a homozygous deletion. Okay, so they have two unfunctional copies of the myostatin regulatory gene, and they can't produce that regulatory protein. And because they can't regulate muscle growth, they grow incredible amounts of muscle, and they're like super jacked. So they're not really good runners, but they're very, very muscular. And so let's do some practice with this little bit of information. So if you could pause your video and go to number 1 and go to this practice in your packet and see if you can fill out phenotype, genotype, gametes, and what is the phenotypic ratio of the puppies. I'm going to pause the video and then we'll pick up with this in a couple, and you can press play when you're ready to look at the answer. Okay, so hopefully you've worked through this. Remember, myostatin regulates muscle growth. So we're going to cross two racing whippets together with one functional and one unfunctional copy of the gene. So when we think about our phenotypes for our dogs, we would label these as racing. If we go to our genotypes, okay, we're doing that M gene. So they have one functional copy that regulates muscle growth and then one non-functional copy that does not regulate muscle growth. And as a result, they're kind of, they're stronger, but still aerodynamic. So M. The gametes that they can make, same way we've been doing this before. Big M or little m, big M or little m. And so if I set up my Punnett square, okay, and so a breeder crosses two racing whippets together, thinking all the puppies will be like the parents. Are we seeing that? No, we're actually seeing a really interesting phenotypic ratio of one non-racing to two racing. Okay, so the non-racing is the big M, the two racing. Okay, so that would be these two individuals right here to one bully. So this one would look like Wendy. So the breeder is only getting like half the number of puppies that they would like to if they'd like to continue raising them to breed. Okay, let's look at the next question. Okay, so Wendy has puppies with a racing whippet. Remember, Wendy is a bully whippet. Okay, so here is Wendy. Wendy is a bully whippet, and she's having puppies with a racing whippet. Genotypes of bullies, little m. Our racing have one functional copy and one unfunctional copy. Wendy has no functional copies of this gene, so it produces tons of muscle because it is unregulated. So if I think about the gametes, big M or little m, here Wendy can do little m or little m, so I'm just going to write little m on this line. If I set up my Punnett square, ooh, oops, excited there. And big M, little m, Wendy can only produce one. Okay, so how many of these puppies will have an advantage when racing? Okay, so if I look here, these are going to be my bullies. These are going to be the racing, so 1/2. Okay, Wendy has 8 puppies. 8 times 1/2 probability of racing would be 4 puppies. And so that would be what you get. What's really cool about this myostatin gene is that this doesn't just appear in whippets. Okay, so here's a picture of Wendy. You can see this in mice. You can see that really jacked mice on the left side. This appears in cattle. There's a sheep right there. So this is a gene that does appear in other animals. There are examples of it happening in humans. It's pretty rare though, but you typically will know that someone has it when they're like this. They've seen examples of like toddlers being able to like do like massive amounts of pull-ups and already have like a six-pack and stuff like that. So it's kind of crazy. So that is incomplete dominance. We're going to pick up with our next example in a couple minutes, but I'm going to switch to a new video so that way I don't run out of time. So make sure you have these filled out, and we will pick up with codominance and multiple alleles in just a moment.